Dear friend, in this module, we are going to see how bioequivalence studies are being conducted for a narrow therapeutic index drug and what are the statistical approaches are used to conduct bioequivalence studies of narrow therapeutic index drugs. What is narrow therapeutic index drugs? Narrow therapeutic index drugs are drugs where small differences in dose or blood concentration may lead to serious therapeutic failures and or adverse drug reactions that are life-threatening or result in persistence or significant disability or incapacity. NTI drugs generally have the following characteristics. Steep dose response curves for both safety and efficacy in the usual dosing interval. They are subject to therapeutic drug monitoring based on the pharmacokinetic and pharmacodynamic measure, and they are generally small within subject variability. Examples are digoxin, lithium carbonate, phenytoin, tacrolimus, theophylline, levothyroxine, divalproxodium, and warfarin. What is the recommendations of Advisory Committee for Pharmaceutical Science and Clinical Pharmacology meeting on NT8? The average bioequivalence limit 80 to 120% is not sufficient for critical dose or NT8. Both requirements for confidence interval should perhaps be narrower, that is 90 to 111%, and include 100%. And we need to carry out replicate studies for NTI. So based on those recommendations, what are the approaches have been developed for NTI? The approaches includes direct tightening of average B limits as Health Canada and EMEA recommended, and the scaled average B approach where B limits are tightened based on the RLD's reference products intra-subject variability. And in addition, if a narrow therapeutic index drug within subject variation is greater than that of the reference product within subject variations, the larger variation in blood concentration may increase the likelihood of therapeutic failure or adverse events. Hence, the intra-subject variability of the test and reference product also should be compared. And also, the approaches evaluated methods and study designs to determine if the generic product has equal or less within subject variability than its reference product to ensure interchangeability within a subject. The recommended bioequivalence study design for narrow therapeutic index drugs are 4-period replicate designs. In this, test product given 2 times and reference product given 2 times. This design will provide the ability to scale the criterion to the within subject variability of the reference product and compare test and reference within subject variances to confirm that they do not differ significantly. The advantage of replicate design is ideally the test product should not be of poorer pharmaceutical quality than the reference product. It means that the magnitude of the test to test variance should not be greater than that of the reference to reference variance. If at all, variance value should be less than the reference to reference variance. This is the typical randomization schedule used for a fully replicated design. It includes 2 sequences, which is TR-TR, and sequence 2 is RT-RT, which includes 4 periods where test product is given 2 times and reference product given 2 times to assess the within subject variability of the test product and as well as the within subject variability of the reference product. The bioequivalence limits of generic narrow therapeutic index drugs are the reference variability is less than 10%, equal or less than 10%. Then the B limits are reference scaled and are narrower than the 90 to 111%. If the reference variability is more than 10%, then the B limits are reference scaled and wider than the 90 to 111%, but are capped at 80 to 120% limits. The proposal encourages development of low variability formulations. And the overall design is reference scaled average bioequivalence for the bioequivalence studies of NTA drugs. This slide presenting the bioequivalence criteria of the narrow therapeutic index drugs, how the 95% confidence upper bound is calculated, and what are the terminologies used in the formula. So for the bioequivalence criteria of the narrow therapeutic index drug, we have seen in the previous slide the criteria 1 is the 95% upper bound should be less than 0. And the criteria 2 will be the 90% confidence of the relative mean of the test and reference product should be within 80 to 123% of the log transform data. And finally, the criteria 3 will be the upper limit of confidence interval for the test variance divided by the reference variance is equal or less than 2.5. This slide presenting a US FDA individual product recommendation for a narrow therapeutic index drug. So this indicates how the study design should be. the scaled bioequivalence limits to the variability of the reference product, and then the comparison of the test and reference product within subject variability also mentioned. So friend, with this, we are concluding this module, which is talking about the narrow therapeutic index drugs bioequivalence studies and the statistical calculations involved in the analysis of endpoints for these bioequivalence studies of narrow therapeutic index drug. Thank you.